Okay, so now, thank you. We now resume the lectures, I'll talk for about 30 minutes. So I want to give you a little bit of an empirical evaluation of these correlation networks applied to genomic data. And I acknowledge right away that much of this work was based on Lin Song's dissertation work and, of course, Peter Langfelder's work. So remember, yesterday I described this study of aging effects in methylation modules. And I just very briefly want to go through it again, but this time I will talk about R commands. And then I will talk about when is hub gene selection better than standard biostatistical analyses. And then we'll talk probably about mutual information and so on. So remember this presentation on aging effects on methylation levels. And remember, we carried out a consensus module analysis. So we had 10 different data sets, and we constructed a signed correlation network in every single data set. And then we found these consensus modules. And next, we related the modules to age. And there is an R function which is called Blockwise Consensus modules. In our R tutorial, we just talked about blockwise modules. You learned how to use it. Later on in the R tutorial, there's a whole section on blockwise consensus modules. But in any event, after we applied this approach, we found this dendrogram. And remember, we found this one green module, this one branch colored in green, where all the genes related to age. And we showed that here that the module eigengene of this module really relates to chronologic age. Remember, very high correlations, particularly in brain tissue. But there is a question you could ask, which is, how do we find hub genes in a consensus module? So let's think about it. So there are really two strategies. The one is to use the consensus adjacency matrix. Remember, when you have 10 different adjacency matrices, one for each data set, and then we form a quote-unquote consensus adjacency matrix, which is yet another matrix. And then you could just define connectivity based on summing adjacency values. However, that approach is unattractive because we always like KMEs, And so therefore, an alternative is to use consensus KME. So there's literally an R function called consensus KME, where you input, for example, your 10 gene expression data sets as a list and the module labels. And then as output, you will get various measures of, in essence, these are weighted averages, Remember, that was what Mike Oldham, in essence, used. Mike Oldham collected, as he said, almost 10,000 brain samples from different studies. And then he got modules in each study, and in some shape or form, he formed an average of the KME values across the studies. And so just so that you're aware, there's this function that does it for you. In some shape or form, it's a weighted average of KMEs where the weight may reflect the sample size, So based on this consensus KME, we ranked the genes in the green module. And we found that these genes were involved in neuron differentiation, nervous system development, and so on. And also, we found that these modules, genes, were enriched with genes that are known polycomb group target genes. Some of you know what these polycomb proteins do. They are very important for maintaining, for example, the stemness in an embryonic stem cells, or they guide cell fate decisions, should a cell be differentiated, and so on. But the good news is that this is already a well-known finding from methylation studies that these polycomb group protein targets tend to get hypermethylated with age, and therefore we can use it as a gold standard for comparing statistical methods, right? Why? Because if you have one statistical method that aims to find age-related markers, and the resulting list of markers is highly enriched with these polycomb group targets, then your analysis was meaningful. If you have another method that leads to a list of markers that is not enriched, you have a bad method. We have a gold standard here. That's very exciting because now we can really compare different approaches for selecting genes. And for example, here every bar corresponds to a different way of selecting genes. And the y-axis reports an enrichment score, in certain ways a log of a p-value in some shape or form. And what we see then is that certain techniques are better than others, you know. Now, that kind of idea was used in this paper from Peter Langfelder, where we asked, when is hub gene selection better than standard meta-analysis, you know? So, let me give you some historical background. As you know, for decades, statisticians have developed very good tools for selecting variables. Keyword student t-test, you know, or a regression model, right? They're wonderful too. And also when you have many data sets, let's say you have 10 different methylation data sets, statisticians know how to deal with it. It's called meta-analysis. For each data set, you get a t-statistic and a p-value, and then statisticians have ways of combining the p-values in an overall p-value. That's meta-analysis. All right, but here you are in a course where we teach you WGCNA, you know. And so you may, of course, wonder, well, what does it buy us? Why WGCNA? Why not meta-analysis, you know? And that's a question I asked myself as well, obviously. And so after many years of research, that's what we came up with. So I should say that I think we solved the question for the special case when you have multiple data sets. Because the truth is, if you have a single data set, it's harder to solve it, I feel. And so that's one question. Let me come to another question, which is the following. So you have a correlation network, and remember we keep talking about intramodular hubs. So genes that are highly connected in a module. But there's also something called whole network hubs. There are genes that are highly connected to all the genes in your network. So if you have 20,000 genes, you can, of course, sum the adjacencies between one gene and all the remaining 20,000 genes. And if that implicates genes with high connectivity, they're called whole network hubs. And there is a distinction, because it could be the case that you have a gene that is an intramodular hub in a small module, small little green module. But in the grand scheme of things, in the whole network, that gene doesn't really stand out in the whole context. And so naturally the question is, should we look at intramodular hub genes, or is there merit in looking at whole network hubs? And a priori, wouldn't it seem reasonable when you have a whole network hub, meaning a gene that is connected to 10,000 other genes? Arguably, that should be a very important gene. But I will show you in a few minutes that this is not the case at all, you know. The bottom line will be whole network hubs should not be looked at in these analyses. And I'll show you the data why. So here then these questions. Are whole network hub genes relevant, or should one exclusively focus on intramodular hubs? Answer is focus exclusively on intramodulars. Next question. Do network-based gene selection strategies lead to gene lists that are biologically more informative than those based on standard marginal approaches? Answer yes, So the whole reason why you are at the course is because many of you have you either are biologists or you have collaborators who are biologists, and they use WGCNA. And why is that? Because the method is very good at learning biology, So, and I'll show you the data that back it up. I do a comparison, the standard method versus network method. So that's the short answer. But let's come to question number 3. So do network-based gene selection strategies lead to gene lists that are more reproducible than those based on standard marginal analysis? And the answer is overall not necessarily. What do I mean by that? All right, imagine you want to develop a biomarker of, for example, for a disease. You want to have a biomarker that predicts whether a person will have a woman has a breast cancer recurrence. Then what do you care about? You want a biomarker, you want a gene that works in every hospital, in UCLA, at Johns Hopkins, and so on. Very reproducible, reproducible in what sense? That the gene expression level of that gene are highly associated with cancer recurrence. That marginal association has to be very tight. And if that's what your purpose is, this biomarker, in my opinion, you might as well use a standard meta-analysis. That's the upshot. And I'll show you the data for it. So we judged different gene selection methods according to 2 criteria. The first criterion measures the biological insights gained. Does the resulting gene list really explain the underlying disease process, the biology? And that's a basic research criterion. Criterion 2 is about biomarker development. So criterion 2 evaluates validation success in independent data sets. And that's relevant when you want to develop diagnostic or prognostic biomarkers. So as a statistician, I could now simulate a lot of data, but they wouldn't convince anybody, right? These are questions of great practical importance. We need practical examples. And here I describe results from 3 different applications. So application 1 was to identify genes that are predictive of lung cancer survival, So we have many lung cancer data sets, and you want genes that predict how long somebody will live. Now, it turns out that biologically speaking, there is a certain category of genes that lends itself as gold standard, which is cell proliferation genes. When as the cancer grows, cells divide, so you need mitosis and cell proliferation genes to be active. So that we know that we want to find genes that relate to cell proliferation. That's the first application. Application 2 is an analysis of aging-related methylation markers. And remember how I told you that we know the gold standard. We know that CpGs that correlate with chronologic age, they tend to be located in polycomb group protein targets. And the third application is to find genes that relate to total cholesterol in mouse liver tissues. We just knew from various publications that high cholesterol levels really lead to inflammation, and so you find immune system related genes in the livers. All right, and so then we had these data sets, and we used 2R function in the WGCNA package. There's actually an R function, I barely talk about it. It's called meta-analysis. So let's say you have 10 lung cancer data sets. This function would do meta-analysis on all these data sets, meaning standard statistical meta-analysis. And then, of course, there's this other R function that I mentioned a minute ago, consensus KME. So you have your modules. It automatically ranks genes according to their hub gene status. And so here are now the results. And let me describe it. So you see the upper panel that which pertains to lung cancer. Every single bar corresponds to a different gene selection method. The bars on the left of this dashed line are various meta-analyses based on, in some shape or form, t-tests. So this is known as marginal meta-analysis. It doesn't involve any modules. It completely ignores co-expression. On the right-hand side, you see bars that correspond to selecting intramodular hub genes in one shape or form from these consensus modules. And in the middle, between the dashed lines, these are gene selection methods where we used whole network hubs, not intramodular hubs. Okay? And the y-axis shows the enrichment score with respect to cell proliferation. Remember, this is, or cell cycle. So these are adenocarcinoma sample, lung cancer, and you can just say which of these methods selected genes that are highly enriched with cell cycle genes. And what do we see? Clearly, all the methods that used modules, consensus KME, huge enrichment scores. And also, there are different ways of looking at hubs. You can use a Stufer method or scale things or rank things. Bottom line is, they all work fairly well. But it is absolutely critical to look at modules. That's how you learn the biology. We all know cancer relates to cell proliferation and cell cycle, but if you didn't know it, then WGCNA would have told you people die because cell proliferation, cancer grows. So it's kind of, you would have learned it. In contrast, these standard approaches that use t-tests, they would have found barely anything, And if some of you study lung cancer, you will probably be aware this is one of the most heterogeneous cancers out there. It's so hard to find any signal in lung cancer, All right. In any event, so clearly WGCNA was the way to go to learn biology. Now let's come to the second application, which is to find age-related epigenetic markers. So here, the story is actually a bit different. So here the standard approaches are still inferior to the module-based analysis, but they work well, right? Look at enrichment scores are still very significant. And that really implies that these standard analyses could detect the signal, and there are probably a dozen publications that found that signal. The standard methods worked. You didn't really need WGCNA. It works better, but you didn't really need it. But I do want to highlight here now the contrast between intramodular connectivity and whole network connectivity. There is no enrichment for whole network connectivity, right? So in other words, CPGs that are highly correlated with lots of other CPGs, they don't relate to chronologic age at all. Totally irrelevant for the question. That's why we make this big deal of look at eigengenes. Which eigengene relates to your trait of interest? It's always a critical step to find the right module to look at intramodular hub genes. All right, and the third application is, of course, to find genes that relate to total cholesterol in mouse liver data. And here again, WGCNA works much better than the standard approaches. People probably, they barely know it, but yes, high cholesterol leads to upregulation of immune-related genes in liver. It's not really that well known why standard approaches don't find it. But if you use WGCNA, you'll find it. All right. So this is, when we look at these three applications, we learn more biology through network methods. But now let's do another, now let's address criterion 2, validation success. Let's say we want a biomarker that relates to cancer survival. We literally want to have 3-4 genes. If they're upregulated, we really monitor this cancer patient and say he's in big trouble. Now here, there's still WGCNA and the intramodular connectivity has an edge. It's slightly better, but not noticeable in lung cancer. Notice that in the aging study, the opposite starts to happen. So here, using the standard marginal approach, totally ignores co-methylation relationships, leads to CPGs that are more consistently related to age. They are more reproducible. So if you want a biomarker of aging, don't use WGCNA. WGCNA teaches you biology. It will tell you it's polycomb group protein targets. They get hypermethylated. You learn biology. But for a strict biomarker of aging, don't use it. And on Friday, I will present a talk about an epigenetic biomarker of aging, which is known as the epigenetic clock. And sure enough, I did not use WGCNA to develop it. I used a different technique. In any event, the third application with the cholesterol levels, again, if anything, these standard approaches perform ever so slightly better than WGCNA, in terms of reproducibility. They just validate better in independent data, And again, we find the whole network hubs, they fail miserably, So therefore, we are very confident in the statement, never look at whole network hubs. So overall, I would say that hub gene status, looking at hubs, teaches you a lot of biology, but if you want biomarkers, you might as well use meta-analysis. And I'll stop with this aspect. Do you guys have any questions at this point? Yes? Yeah.
So Steve, in our data sets, several data sets, I find that differentially expressed genes are highly enriched in the gray module.
Yes.
So along the lines of what you just talked about, my thinking about that is that if you're a hub, it might be harder to modify, even in cancer or in biological situations.
Yes.
And by the same token, if you do change a hub even a little bit, that's a biologically more meaningful event than if you change a lot some peripheral gene. So what are your thoughts on that?
Yeah, so to repeat for everyone, so sometimes it turns out that the gray module is the most significant module, and gray denotes the background. It's not really a module, it's an artificial module. And this is always puzzling, in some shape or form, because the gray module is arguably poorly defined because its eigengene often doesn't summarize the module, the low proportion of variants explained. But it does happen that for some strange reason, it's very highly associated with the trait. And then I'm always quite perplexed what to make of it, And so the comment was, which is a good comment, was, well, maybe it could be that hubs are really tightly, intramodular hubs are actually tightly regulated, and so there's not much leeway to be disrupted, whereas these genes that are not in modules, if anything, they could actually relate to your disease because they're not as tightly regulated. I like that interpretation a lot, so by all means, that's so just go with it. I would just then say, oh, what is known about the gray module genes? Why are they in the background? Do a functional enrichment analysis and just use the standard gene significance measure, right? You have a disease and you say, so which genes in the gray background really relate to the disease? Why do they do? I would just go with the data, you know. Yeah, so I would say in many applications, looking at modules really teaches you a lot of biology. I think you've seen lots of case studies, but with disease, who knows? There could be a few genes that drive the disease, and they don't operate in the context of a module. You know? Yeah. All right. Other questions or comments? Yes.
Can you comment on the significance of the second point, like why it is not, or why the intramodular hubs are not the good validating candidates?
Yes. All right. So before one does a statistical analysis or any research, you always need to say, what am I after? You know. So when you are in basic research, let's say you have a disease such as autism, and we don't quite understand why do some people get autism, others don't. It's a developmental disorder, but we don't know the biology. In my worldview, in that context, you are at the stage of learning biology. Therefore, network methods can make a big difference. They may pick up subtle signals, they amplify the signals. And how do network methods amplify the signal? Because they look at the module, right? You throw away all other genes, you look at these genes that are co-expressed with other genes. They allow you to find cell types that are involved, pathways that are involved, and so on. So in certain ways, a module-based analysis is very valuable. However, let's now assume a different disease. For example, let's say you want a biomarker of aging. You want to take somebody's blood and you want to say how old is that person, you know. In certain ways, suddenly you no longer want to learn the biology. You actually don't want to, you don't care that much why that person is old. Well, you say they are old because they just had their 80th birthday, you know. That's you don't really care all the pathways. All you want to know is a very accurate predictor of age. And in this context, you suddenly need a whole different set of tools. That is known as, the label would be supervised machine learning methods. You need machine learning tools that predict things. You need prediction methods, or sometimes known as classifiers. You just don't need network methods, you know. And and so in this sense, if you keep, so in this application where you want high predictive accuracy, in my opinion, networks may not be that valuable, you know. Yeah. No, personally, I don't think that's the difference. I don't think directed networks would make a big difference. I mean, so I want to position these statements as my opinion, you know, because you will go to other researchers and they will counter these arguments, you know. They will say, oh, how dare you say that? That makes no sense, you know. But I say it because I analyze data for a living. I have 20 years of experience, and I develop biomarkers. I develop WGCNA. I do it all, and in my opinion, just from the school of hard knocks, you know, turns out that there's a dichotomy here, you know. Yes, Morgan?
I think it could be because these methods are more unsupervised.
Yes.
And if you were somehow able to incorporate the networks into a supervised learning
Yes.
method, maybe they'd be better predictors.
Yeah, definitely. So WGCNA should be considered as an unsupervised machine learning method. I mean, it's not clustering. I mean, clearly you see clustering is a big component, but there's way more to it. Network concepts that I didn't teach you yet. There's connectivity. There are things like module preservation statistics that we'll talk about. And really topology matters in networks. And but topology doesn't matter for biomarkers, in my opinion. So when you want a predictor, you are really in the realm of supervised machine learning. And as Morgan indicated, there may be a room to develop a hybrid where you tweak networks so that they are semi-supervised or so, and that could well be the case in the spirit of partial least squares analysis, So yeah, but I have never seen anyone present a convincing method for predicting an outcome based on networks. So a method that is drastically better than off-the-shelf supervised methods such as random forest, penalized regression, Just be aware of it, Okay, please.
I think that biomarkers are not very well detectable by network methods does make biological sense, because normally you see, for instance, the biomarker is very often the endpoint of some process in the cell. Take, for instance, interleukin 8, which is a classical biomarker for inflammation, which is secreted by endothelial cells, and we have seen this with gene co-expression network analysis, that within a module which is defined by the G-motor positive regulation of inflammation, that interleukin 8 and others, interleukin 6 and so on, which cause endpoints, they are not on the hub genes.
Yes. Yeah, I assume everyone heard that, So that's a good example, Yeah. Yeah, I would say let's make a 10-minute break and then we shift to another speaker, and thanks.